Welcome to UC Davis Live COVID-19. I'm Satirius Johnson. In this week's show, we'll be stepping into the lab to talk about some of the work that's going into making vaccines for COVID-19. Many efforts are underway around the world to develop a vaccine in record time. So how do scientists go about testing a vaccine like this? What lessons do we have from previous research on other vaccines and other viruses? Here to help us get answers to those questions are Kristen Grimsrud, Assistant Clinical Professor in the UC Davis School of Medicine, Department of Pathology and Laboratory Medicine. She's especially interested in taking discoveries from basic lab science and translating them so they can be applied in humans. We also have Dennis Hartigan O'Connor, who's an Associate Professor in Medical Microbiology and Immunology at the UC Davis School of Medicine and a core scientist at the California National Primate Research Center. He studies the development of the human immune system and how it interacts with infections. Thank you both for joining us today.
Thank you. I'm happy to have the opportunity to be here with you today.
So we will be taking audience questions. If you're watching us live on the live stream, just leave them in the comments and we will try to work in as many as we can. So Dr. Grimsrud, you're also a veterinarian and you work at the UC Davis Mouse Biology Program. How are mice and rats contributing to understanding or developing vaccines for the new coronavirus?
Yeah, so mice are a great research tool and they've been used on a global effort to study COVID, and people are utilizing them to look at different genetic susceptibility, to look at different treatment options, as well as vaccines. And there's a lot that is being done to genetically modify these mice to make them better research tools so we can really translate the data that we get in the lab to what can be implemented clinically. Rats have a much more limited use because they don't have quite as many options for genetically modified rats, but they can still be of value to look at early pilot studies and quick screening studies to optimize vaccines and some quicker research before going into either larger species or humans.
Do rodents get COVID-19 the way that we do?
Yeah, that's a really good question. Rodents that are not genetically modified, or at least mice and rats, do not get the clinical symptoms as humans do because they have different genetics behind their ACE2 receptors and some other genes. So in order for them to be susceptible to the disease itself, we need to genetically modify them so we can allow that virus to get entry into the cells, as well as see the actual hyperimmune response that we're seeing in humans. And so the basic mouse or rat do not, but other species such as some hamsters, ferrets, and primates do get more of the clinical disease. So there's a stepwise approach on what animal models are best for the research purpose.
So when you're looking at mice and rats, as far as a vaccine goes, what are you looking for when you're testing a vaccine?
Yeah, so work we're currently doing at the Mouse Biology Program in partnership with Vernari is using rats to look at early vaccine studies. And so what we're trying to do is just look at whether or not the animal's getting a good titer and what that immune response is. So we're not challenging them with infection, but we can do some early screening to optimize the vaccine itself in terms of what adjuvants or what antigens are within that vaccine before moving on to larger species such as the primate, which are the basic steps that really need to be looked at for vaccine efficacy before getting into human clinical trials.
So it's basically like a first step before you get
exactly
to
and we're in a hurry to do this, and research takes a lot of time. People want a vaccine now. And so we're trying to make this very quick in some smaller species before going into larger species.
Because smaller species, their lifespans are shorter, everything else, it's just a quicker process.
Yes, it's just an overall quicker process. Right. And that way we really can optimize the vaccine before they get challenged as well. So it's just a quicker approach. So we really want to test the best possible options, and it gives us a quicker screening to be able to do that.
Now, you've been involved with testing vaccines based on a skin patch. What's different about that approach as opposed to a vaccine that's injected into, say, an arm or something like that?
Yeah, so we started working with Bernardi on this at the early stages about 4 years ago, and it really has some great utility in that it's transdermal. So it's a simple patch that people can even apply to themselves at home. There's not the pain that people have with injections. It doesn't need to be refrigerated. So the goal and the concept behind this is that it could really change things globally. You could send it in the mail. It can be sent to third world countries. It can be sent to people at home so they don't even need to come into the clinic. And it can be mass produced quickly. And so that's the concept behind that. And there's a good immune response in the skin itself. And so you're actually able sometimes to get even higher titers with a lower dose, or the same dose, but even a greater effect with the transdermal approach.
Is this skin patch approach something that's been tried in vaccines for other diseases?
Yeah, so Verndari, as well as many other groups, have been working on this patch approach for many years now. And Verndari themselves have done it with other things such as influenzas, what we started with, but outside of our program, they also work with it for shingles and rabies. Other groups are even using it for cancer vaccines. So there's a lot of utility behind this approach.
And as a veterinarian, what do you bring to this kind of work that's different from, say, a physician who treats people?
Yeah, so from the veterinary standpoint, what I bring is that I can work with researchers to choose the best animal model for the goals of their study. And then I'm actually a doctor for the animals involved in the research. So we want to provide them the best possible care, make sure they're in a low stress environment and healthy, and make sure we're only using the absolute what we need to get the research objectives accomplished, and not use more animals than need be, and make sure they're in a safe, healthy environment throughout their study.
Again, we are taking questions. So if you're watching us on the live stream and would like to ask a question, please just drop it in the comment section and we will try to work it in. Dr. Hartigan O'Connor, I understand you've previously worked on HIV vaccines. How did you pivot from that to working on COVID-19?
Well, HIV vaccines are one of certainly the greatest immunologic challenge, and maybe one of the greatest scientific challenges of the last 30 years. So thousands of new vaccine technologies have been developed in the context of HIV. HIV is also a particularly difficult vaccine target, and so many of these technologies are the most powerful approaches that we know of to stimulate immune responses in people. And so in terms of the pivot, it's actually sort of a, as soon as the pandemic began to grow and people realized that it would become a pandemic, actually you start to get calls from colleagues and grant making agencies and others who want to know if these technologies that have been developed over the years are the key to helping in the pandemic response.
Well, we're still not, well, as you kind of mentioned, it's very challenging to develop a vaccine for HIV. We're not there yet. How promising are the prospects for a COVID-19 vaccine?
Well, the biggest fundamental difficulty in an HIV vaccine is that we don't know of any person ever who's developed an immune response that can clear an HIV infection in human history. So that presents an enormous challenge, because in HIV we're forced to do something that we're forced to try to do something that we don't know if the human immune system is capable of. But in contrast, we know that people do cure themselves of COVID-19. It's the most common outcome of the infection. And so you've got a big head start right there, because you can look at people who have been able to cure themselves and those who have more trouble, and try to figure out what's the key responses that you need to induce. I think the problem is that not everybody cures that infection. We know that the coronavirus has learned to live with the immune response against it. So even though that response happens sometimes and is sometimes effective, and that's very encouraging, we also know that the virus and bad disease can coexist with a very good, powerful immune response. So we have to find the right response for a vaccine.
Why are rhesus macaques a good model for studying vaccines or even treatments for diseases like HIV or COVID-19?
You need two things to develop a vaccine. You need an immune response that's appropriate and similar to what you're going to get in people, and you need a disease that's similar to what you're going to see in people. And then you have to match the two up. And so rhesus macaques kind of have that perfect combination. They're susceptible to the infection. And so they develop some symptoms and some disease that are similar to what people with COVID-19 can have. And they also have an immune response that's similar to humans, not just genetically, which is important. It's not just that they're closely related to humans, but rhesus macaques are also highly variable. We have young macaques, we have old macaques, we have macaques with diabetes, we have macaques that really model the spectrum that you're going to see when you try to protect human beings. And that's the real challenge for a vaccine, is learning how to protect the breadth of the human population and not just the youngest, healthiest people.
You mentioned diabetes. I know that you recently got a grant to work on a COVID-19 vaccine for people with diabetes. What's the issue there? What's different? What is different about a person with diabetes who gets COVID-19 or could get it, as opposed to somebody who doesn't have diabetes?
We actually don't know what the key difference is. We know that there are certain groups of people, and it's primarily people with comorbidities like diabetes or people who are older, who tend to do worse. And unfortunately, those are the same people who have often difficulty responding to vaccines. They either mount a lower response to the vaccine at the beginning, or that response tends to decay more rapidly. And so the issue is that precisely the people that you're most anxious to protect are the ones that in a way are hardest to test out in animals. Because most common thing in an animal study is to study young, healthy animals because they're the most available. And so we're trying to address it by taking some of those HIV vaccine technologies that have been most effective in terms of overcoming those specific immunologic challenges that we know older people have, and both using those vaccine approaches and also testing those approaches in diabetic animals or older animals.
Maybe this might be a good time for either one of you to maybe remind us how a vaccine works. How exactly does a vaccine work?
Well, maybe I'll start, Christian, then you can jump in. There's two major arms of the immune system that we think about. There's antibodies and T cells, and most of the vaccine development work in COVID-19 is focused on antibodies. And what antibodies are the simplest kind of protection you can imagine, in that they are little proteins that are made in the body that literally stick to the outside of this invading pathogen and prevent it from working just by coating it in an innocuous protein. And so the job of the vaccine is to make as many of those antibodies as possible. You can also engage the T cell response, which is a key part of the HIV-related vaccine effort. And that's something that's received less attention so far, but it's what we'll turn to next if the antibody-based approaches don't work. And those fight the virus after it gets inside to a cell. And there, the goal of a vaccine is the same. It's to create as many of those protective T cells as you possibly can, so that you fight the virus in as many infected cells as possible.
Yeah, and to add to that a little bit, the vaccine, in terms of optimizing how you're designing it, is a combination typically, depending on your approach. So, for example, what we're working with right now, it's an antigen, so it's a protein, it replicates a piece of the virus, and then we have an adjuvant combined with it, so that helps us build a stronger immune response. So by delivering the vaccine, either through the patch or an injection, that's when the body then recognizes it as a foreign that shouldn't be in its body, which is what we want. And then that's when we want the immune system to take over and try to clear the body of it. And so during vaccine research, we're trying to find the best combination, whether it's a different dose or a different type of antigen in combination with the adjuvant, in a system, a delivery system, to optimize that immune response.
If I could just add maybe even one more point, I think Kristen and I are both most focused now on these subunit approaches, or this idea of introducing just a little piece of the invading pathogen. And that's because that's the newest approaches and the highest technology approaches and the ones we're both interested in. The older technique is to just take the whole pathogen, the whole virus, and inactivate it with a chemical, and then inject that, and hope that the body mounts a response. And that's something that's actually could be effective in COVID. And there's a Chinese vaccine that's taking that sort of or lower tech approach, but that's not the focus of any major vaccine efforts in the U.S.
We do have an audience question. Lyric on Facebook is asking, currently, companies are going straight to humans to develop COVID-19 therapeutics and vaccines, bypassing animal studies, basically since there isn't a SARS-CoV-2 challenge model currently, although there are other COVID models, SARS-CoV-1 and MERS. There is also the urgency of the situation. So what are your thoughts on the current trend of going straight to humans? Should we require more animal studies first, or just go straight to humans?
So when we're talking about treatments, we're also looking at drugs that have already been used in humans. So they have already gone through a lot of time, both animal and humans, to show safety. And that is the big key with both vaccine and pharmacology research. We need to make sure that what we're administering isn't going to have so many side effects, we're actually causing more problems. That's part of why we do the vaccine research as well. We can have a hyperimmune response, we can have adverse reactions. So the drugs that are currently being used, like recently dexamethasone, we've been using that in human and veterinary medicine for a long time. So we do have some background knowledge of its safety. Dennis, you can share more thoughts on that as an MD.
Yeah, I think there's actually a continuum of knowledge. So it's not that any of these technologies are completely untested in animals. All of them have been in animals, maybe for other pathogens, maybe for the first iteration of the SARS coronavirus 1. And so the FDA, before letting anything go into humans, does have a lot of information about safety in animals that may not be precisely the vaccine that's being tested, but that's very, very close. And so that safety question is addressed. In people, the biggest problem you have is that most of the people you vaccinate, in fact, you hope by far the vast majority will never be exposed to the virus. So for many of your vaccine participants, you won't learn anything because they won't be exposed to the virus, and you won't find out whether they're protected or not. So that ability to take an animal who's been vaccinated and expose the animal to the virus and find out for sure if that animal is protected or not is valuable information, whether you do it before you start your phase 1 clinical trial or afterward, because it still informs your understanding of how good the protection is.
Danielle, who's watching on Facebook, is asking, can we talk about the challenges of developing a vaccine to COVID-19, given that it seems there are 2 strains, and given what you know about the genome stability of COVID-19?
Yeah, that's an important point. And one of the great things about the coronavirus, in contrast to HIV, is that even though there are potentially 2 strains, in general, the sort of cloud of mutations and variants is tiny compared to something like HIV, where any vaccine target at all that you want to consider in HIV is something that can be and will be mutated. So whatever target you pick, the target's going to change on you. So that's very different in coronavirus. And with the coronavirus, these 2 strains are defined by a single mutation, or the putative 2 strains are defined by a single mutation. And so for many vaccine targets, that mutation is just outside of the vaccine target, and it's not needed for the vaccine to work. So certainly it'll be important to test these vaccines against strains with different properties. But the fundamental problem of making an immune response against a certain specific restricted part of the virus isn't affected by the strains that we know about so far. Right. So far, the variation is pretty limited.
Right. So basically you're saying that the difference between the strains, the possible 2 strains, is so small that the vaccine would work for either of them at this point.
We think that's right. Yeah, we think that's right.
If we test the vaccine in rats or monkeys, how confident can we be that it'll work in humans? Like, what do you look for?
You want to start in the small animals, Kristen?
Yeah, so what we're mainly looking for is we're taking serial blood samples to see if they're developing a titer, so if they have that antibody response. as well as we're looking at biomarkers for the inflammatory response. So that tells us that the vaccine got into the body and the body responded in the manner in which we were expecting it to. And then from there, Dennis can speak more to the work done in the primates and other things on what they're looking for once they're challenging the primates with the actual virus.
Yeah, so the key is to match the immune responses that Kristen's talking about to how much protection you get from that given level. So if you have a antibody titer, say 1 to 10,000, then you have to know if that's enough or it's not enough. And the only way to do that is to challenge animals who have different levels of response and find out how much of that response do you need before you can be confident it will work in people. So that's a big part of the reason for studying these questions in animals, so that we can figure out when we see a given response in a human clinical trial with a person who hopefully and probably will never be exposed, do we nonetheless expect that that response is protective?
I would guess that you would also maybe what kind of an idea, I mean, you talked about the efficacy of the vaccine. Can we also get a sense of the safety, as the human safety, when we're testing in rats or monkeys?
Absolutely. Yeah, that's, that's sort of the first baseline step that you do in terms of justifying a clinical trial to the FDA, is to summarize all the experience that you have with this vaccine or the vaccine technology. In the case where you've developed a vaccine so rapidly that you haven't tested in animals, you nonetheless will have tested versions of that technology in animals. And so the first thing you have to do is to summarize for the FDA everything that you know about its safety. And everything that's known about its safety in humans as well from prior trials that may have gone on. And I don't think anybody who would feel confident taking a completely untested technology to people.
We have had, of course, vaccines for many diseases, things like measles and polio for a long time now, but we're still waiting for a vaccine for HIV, as we've mentioned. For example, what makes some vaccines harder to create than others?
Well, there's certain things that we know a lot about, and we know exactly how to do them. So the simplest immune response that we know how to create is antibody responses. We've been studying these responses for a long time. We have 100 different technologies or more to elicit them. And so the questions become, do you know what you need to generate antibodies against? And do you know how much of those antibodies you need? And so with a new outbreak, with a pandemic, with an epidemic, we don't know those things. We don't know what's the key target in the virus. We don't know how sensitive the virus is to inhibition. And in the case of certain respiratory viruses, there's another issue, which is that the wrong kind of antibodies or the wrong level of antibodies can sometimes make the disease worse. So that's a tremendous concern with all respiratory infections, particularly because some of the best examples of that phenomenon of antibody responses that make the disease worse are in respiratory infections. So what makes some vaccines harder than others is basically the target and how much you know about the target. If you know what you need to do, and if the virus is particularly sensitive to what we're able to do, then it's easy. And there's a lot of diseases like that, and coronavirus can still be one of them, maybe one of them. The big challenge we have in coronavirus that makes it harder is we don't know enough about it because it's so new.
And I would imagine that it's very tricky with this virus, because one of the things that is killing people is that it seems like the body is responding with inflammation, which fills up the lungs with fluid. And
that's exactly right. And that's the kind of
immune response, right? Is that what you're trying to do? You're trying to fine tune the exact kind of immune response?
That's exactly right. Well, it's certain that you can have the wrong kind of immune response. And what we have to be very careful of is that none of our antibodies cause that kind of response. None of our vaccines cause that kind of response. Right now, we don't think that's a huge danger. It seems like the best evidence is that the vaccines aren't going to cause that to happen. But that's one of the things we're on the lookout for.
We have another question from a viewer, Duani, who's watching on Facebook, asks, what challenges might a person who is immunocompromised face? Would their system be able to effectively respond to the vaccine? If not, what would your approach be in this case?
Kristen, I know you were going to say something a minute ago. Do you want to take that?
Oh, well, a moment ago, I was going to add to the previous question that the other challenge is the mutations that we discussed and trying to make sure we, target a vaccine for the conserved regions. With regard to the current question, there's always more concern with people that are immunocompromised, that they're more susceptible to the disease. We have a number of immunocompromised animal models, and the mice has a lot of value in also looking at research behind that to better understand to what degree immunocompromised people may have a different in response and effect. And I think Dennis can speak to that a lot more from his experiences with concerns for humans that are immunocompromised.
Yeah, I mean that immunocompromise is like most human conditions, a continuum. And so one of the key questions in clinical trials, and one of the key differences between the small animal models and the larger animals, is that in the larger animals, you'll get more variability in that continuum from immunocompromise and failure of the response to, a strong, robust immune response. And in humans, you'll see an even greater response as you start to vaccinate millions of people. You'll see an even greater variation, rather, as you start to vaccinate millions of people. And so I think it's a cutoff. The better your vaccine technology is, the smaller the fraction of people who are immunocompromised who aren't going to respond. And then the worse your vaccine technology is, the larger that segment that won't make it is going to be.
How hopeful are you that we'll have a COVID-19 vaccine that works for most people?
I think it's very important the way you asked that question. You said for most people, that gives a specific cutoff of 50% of people. I think the part that question doesn't take into account is that the protection is going to be transient. It's going to have a certain time limit. And so I'm very confident that we'll have a vaccine that works in 50% of the people on the day you finish vaccinating them. So I have no doubt at all. The vaccine technologies that are being tested are powerful enough to make immune responses that we have great evidence are going to be protective on the day that you finish that vaccination regimen. Question is, a month later, 3 months, 6 months later, will you still be protected?
I think part of that too, and what we're looking at now is, there's some vaccines you can give them once and you're good to go. There's others that need boosters. And so you might need to do a series of vaccines, and what that interval is to really give a good response. That's what we're working on and trying to figure out. And then there's a number of vaccines where we might need to revaccinate twice a year or once a year, whereas others can go many years. And so these are all questions that we're looking at. And then we're also thinking about, all right, do we need to optimize the vaccine each season as the vaccine mutates, kind of like what we're dealing with influenza. So it might be that the vaccine helps us decrease the severity versus completely eliminate it. I think from what I've seen thus far, we have great potential to do good work to get a good vaccine for the majority of people, but it's going to take some fine tuning probably.
We have another question from Lyric, who's watching on Facebook. It's been said that the second wave for COVID-19 will be during the next flu season. What complications do you see when a patient has both COVID-19 and flu viruses at the same time? Can you get chimeric viruses? First, let's define what a chimeric virus is.
Chimeric virus would be like a hybrid of the two, a mixture in which the virus would carry segments of both the flu virus and the coronavirus. And that would be extremely unlikely, because actually viruses are a little bit like people in that, or a little bit like species in that similar species are able to, to have offspring and recombine and create new variants, whereas different species aren't able to do that. And in the same way with viruses, two coronaviruses can easily mix their genetic material. And in fact, that's how we think that this coronavirus evolved, from a mixture of bat and pangolin viruses. But coronaviruses and influenza viruses are actually quite different, and it's hard to imagine that they could recombine. So I don't think that specific issue of a chimeric virus is the major problem. It's more the problem that there are going to be people who are burdened by both infections and who might have been able to contain one, but being infected with both is going to make them vulnerable.
And basically, if you have both, I mean, having a vaccine or some sort of therapeutic, you're basically battling both, right? You have to be battling both separately.
Yes, and it's also the case that that one viral infection will transiently make you more susceptible to another one because it cripples your immune response for a brief period. And so you can imagine that even a person who might have been protected before becoming infected with influenza then maybe wouldn't be protected in that period of a week or two after infection. So we don't know that information for these two viruses, but yeah, essentially this is 2 hits is going to be worse than 1 hit.
So just to wrap up, how long do you think it'll take to actually get a vaccine that will get us to a place where, we can start, feeling comfortable reopening and getting close to back to normal?
I'm
not sure if I want to go on a limb first or you should, Kristen.
I can do my default answer, right? So I did a safe answer, and that as many have seen on the news and media, I think it is practical to think that we could have a vaccine in winter, after the new year, potentially. Of course, that can vary depending on how the research goes, how the earlier trials go, but a lot of people are working really hard on this and they're already making great strides. So I do think it is doable to have within the next, year time frame. But as we keep saying, it's not necessarily going to be the magic bullet, and it is going to take some time to learn about human responses, especially once you're in larger populations and people are interacting more. So I think everyone will still need to be cautious on their social interactions and not just think that everything's going to go back to normal.
Yeah, I think there's 2 parts to the answer too. I think one is, as we've discussed, there's sort of a continuum of vaccines. We'll have a vaccine, and the question is, will it be good enough to protect most of us? And then in the future, we'll have better vaccines. So there's not going to be just one vaccine, I don't think. I think we'll have something in the next year. But the second part of the question is, or the second part of the issue is, how long does it take to roll that vaccine out to enough people to have an impact on the pandemic? And that's something that depends partly on force of will and political will, but it also depends partly on the technology. Some of these technologies have never been made at a scale of hundreds of millions of doses. Most of them haven't been. And so it's a real scientific question as to how possible it is to make some of these vaccines at scale. So I think the idea of having an effective vaccine that's going to make all our problems go away in the next, by the end of the year is low. But having a vaccine that works and begins to allow us to relax our restrictions, yes, I think that is possible.
Well, this has really been great. And I have to tell you, we've been getting a lot of messages of support and gratitude for all the hard work that's being done to help the research and move us toward a vaccine. And that's mostly coming on Twitter. So thank you from our audience, and thank you from me for your time and for all the important work you're doing.
Thank you.
Thank you.
Kristen Grimstrud is Assistant Clinical Professor in the UC Davis School of Medicine, Department of Pathology and Laboratory Medicine. And Dennis Hartigan O'Connor is an Associate Professor in Medical Microbiology and Immunology at the UC Davis School of Medicine. Every other week, we are hearing from researchers from across the UC Davis campuses who are working on vaccines, therapies, and medical devices, tracking the virus and looking for solutions in unexpected places. On the next UC Davis Live COVID-19, we'll be looking into the latest on what we know about how COVID-19 spreads. This, as most of the country has started reopening to various degrees and as we head into the 4th of July weekend. Until next time, I'm Saturius Johnson, and this is UC Davis Live COVID-19.